Hi, my name is Bob Bowden, and I'm a plant pathologist with the USDA-ARS in Manhattan, Kansas, and I'm stationed at Kansas State University, where our wheat genetics research group has been working for many years, identifying and developing sources of resistance to all three rusts and releasing them. And breeders have often taken these up and used them, but unfortunately have often deployed them singly or in small stacks so that it really hasn't been sufficient, and we've seen many good resistance genes be defeated by new races of the rust pathogen. And so this is really frustrating, and it's really a shame because a lot of time and money goes into development of those lines, and having a short commercial life is just not cost efficient. So we need to have better methods of gene stewardship, especially on these expensive new resources. So when UG99 emerged as a new threat, we decided that we were exclusively going to pyramid our germplasm lines to try to protect the resistance genes. And so we've developed several methods of rapidly stacking resistance genes, and we've been successful. And we now have lines that have 6 stem rust resistance genes, 4 of them major genes, 2 of them minor genes, and they are in good backgrounds, high yielding backgrounds, and so we're very hopeful that this will result in enhanced durability of the components of the resistance genes in those pyramids. And what excites me about the future are some of the new technologies for large-scale cloning of rust resistance genes from wild relatives of wheat. It has a potential to really increase our arsenal of rust resistance genes, and also stacking them in cassettes to achieve even higher levels of durability and gene stewardship. So the future, I think, is really bright for durable rust resistance in wheat.
My name is Yue Jin. I'm with the USDA-ARS Cereal Disease Laboratory located in St. Paul, Minnesota. I'm also adjunct associate professor with the Department of Plant Pathology at St. Paul campus of University of Minnesota. The Cereal Disease Laboratory at St. Paul, Minnesota has historically played a fairly significant role in understand rust populations, awareness dynamics in particular, and also help United States as well as developing countries in develop rust resistance. Since the lab established has a very close collaboration with CIMMYT in developing varieties. My role in the DRW project is somewhat multi-fold. We carry several objectives of the DRW. One particular area is in the gene discovery. Try to look for wheat relatives, wild species, and try to identify UG99 resistance from those relatives, so we can introduce those new or potential useful genes into cultivate wheat or barley or durum. Success of the project, overall project, in my opinion, so we deliver effective control strategy to control those diseases. In our opinion, the development and the utilization of resistant varieties is the most economical, efficient way to do that. The BGRI project, I think, is one of the best examples how international collaboration can move things in a cohesive, collaborative manner.
Good morning. My name is Mary Guttieri, and I work for the USDA Agricultural Research Service. I'm based in the Hardwinter Wheat Genetics Research Unit, which is located at Manhattan, Kansas, square in the middle of the United States. I am fundamentally a wheat breeder. I execute pre-breeding and discovery projects for the benefit of the Central Great Plains region. I'm honored to be among the recipients of my agency's award for stewardship from the Borlaug Global Rust Initiative. And I was asked to explain how stewardship became important to me. And what I want to tell you about is my experience prior to being a wheat breeder. I worked for 8 years in the discipline of weed science. And in my work, I characterized mutations in weeds for herbicide resistance. And I also conducted herbicide tolerance and efficacy trials to bring new products online for the benefit of our growers. In this work in weed science, I saw firsthand the collapse of two very important classes of herbicides, the ALS inhibitors and the ACCase inhibitors. Despite the dedicated efforts of the research and extension community, safe and effective selective herbicide products were rendered useless by a fundamental lack of stewardship. At the same time, I saw what was required to bring new modes of action online, and I saw what happened when they weren't protected. Now, as a wheat breeder, I see what's required to bring new genetics online for our growers, new genes for host plant resistance to diseases. And at the same time, I'm seeing what happens when those genetics are not protected. My particular role in our agency's team seems to be the one who says, yes, we can. I find underutilized tools like the MS3 trait for dominant male sterility that have really dramatically improved our crossing throughput and reduced our labor requirements. And I couple these with high throughput marker selection technologies to bring forward some pretty complex deep gene pyramids for host plant resistance to diseases. And to bring these into the testing pipeline so that they can then be advanced into breeding programs to benefit our growers. So I'm grateful to be part of the USDA effort and part of this extraordinary team. And I want to express my gratitude again to the Borlaug Global Rust Initiative for this important recognition. Thank you.
Hello, this is Harold Bockelman in Aberdeen, Idaho. My role in this research has been as facilitator. I'm the curator of the National Small Grains Collection. As such, we have provided some of the germplasm that has been used in this stem rust research. The National Small Grains Collection has a large, diverse collection of wheat germplasm totaling over 50,000 accessions, including triticum species, agelop species, cultivated, primitive, and wild relatives, all useful for wheat improvement. My role as curator over the past 30 years what has inspired me has been the interactions with scientists, as I get to see sort of the beginnings of research that is going on around the world. Thank you.
My name is Stephen Xu, and I work at USDA-ARS, Edward T. Sheffer Agricultural Research Center in Fargo, North Dakota. From my plant breeding class in college in the early 1980s, I first learned about the grain revolution from Dr. Bollock's work and the gene-to-gene model from Dr. Flor's work. Then, in graduate schools, I became familiar with the achievement and the impact of the research on wheat genetics, cytogenetics, and gene integration conducted by Dr. Kihara, Shears, McFadden, and other pioneer wheat scientists. Those accomplishments have greatly inspired my research as a wheat geneticist. My role on the USDA-ARS gene stewardship team to identify stem rust resistant gene and associated molecular markers, and then transfer the gene into adapted durum and bread wheat germplasm. In the past 10 years, my lab has identified and transferred more than 10 unique UG99 effective genes, mainly from wild relatives of wheat. We have made the wheat germplasm carry these genes available to various breeding programs globally. Thank you.
Hi everyone, my name is David Marshall with the Agricultural Research Service of the United States Department of Agriculture. I want to thank the Borlaug Global Rust Initiative for selecting this USDA team to receive the Gene Stewardship Award. I've been breeding wheat now for about 40 years, and my role in this USDA team is to breed high-yielding, well-adapted winter wheat germplasm having resistance to rust, mainly based on partial or minor genes, but also overlaid with several different major genes, specific major genes for resistance. I also serve to evaluate the U.S. nursery of winter wheats every year in Njoro, Kenya. The first year of evaluations was in 2005, and it took us several years to learn how to grow winter wheats at high elevation near the equator. I think the key to gene stewardship is to incorporate as many minor and major genes in different combinations with each other and in different genetic backgrounds. That will give us a wide choice of material to move forward for germplasms and varieties, while at the same time it allows us to avoid the problems of genetic uniformity. For me, the most inspirational accomplishment is to see the smile on the face of the farmer, the miller, the baker, and finally the individual eating that piece of bread. I thank you very much.
Hi, this is Tyler Gordon with the USDA-ARS National Small Grains and Potato Germplasm Unit in Aberdeen, Idaho. My contribution to this team is preparing U.S. wheat and barley entries for shipment to Kenya and Ethiopia stem rust screening nurseries so they can be tested for UG99 and other East African races of the stem rust pathogen. What motivates me to do this work is the need to find unique sources of resistance, which we're trying to do by screening the winter and spring land races from the National Small Grains collection against these races of stem rust. Additionally, wheat is an important commodity. It provides roughly 20% of the calories for humans. And so finding sustainable ways to produce wheat and reduce the impacts of growing wheat is really important for agriculture production. Thanks very much to the BGRI team for this award.
My name is Matt Rouse. I work at the USDA-ARS Cereal Disease Laboratory in Minnesota. My role on this team was really three things. First, to couple stem rust resistance genes on the same chromosome arm, so like SR9H and SR28. Second, to backcross those linkage blocks into advanced wheat breeding lines. And third, to deliver the germplasm now with multiple stem rust resistance genes to collaborating wheat breeders. I'm driven by the ARS mission, not only to conduct research to address agricultural problems, but also to transfer solutions to those problems to our stakeholders.